Today I will explain you the cell cycle control. How the cell cycle control of cell cycle. The course code, as you know, is BBBMBT1002, and so now I'm going to start. And in the previous classes I have just explained you the cells, about the cells, its organelles, and then cell cycle, then mitosis, meiosis. So you know about all these. Today I'll be discussing how the cell cycle is controlled. Because we know then in every part of body there is different stage of cell cycle, and the cell division is controlled. Some of the tissues can control, some tissues can some of the cells tissues are having the cells which can divide at very fast rate, and some of the tissues the division of cell is very slow, and some of the cells do not divide. So how all these things are controlled, that we are going to discuss in the control of the cell cycle. Let's come to next slide. So this is slide is showing how the cell division is coordinated. Now this is coordination of cell division. A multicellular organism needs to coordinate cell division across different tissues and organs. This is critical for normal growth, development, and maintenance. How this is performed? What are the factors which are controlled by the cell division control or cell cycle control? They coordinate timing of the cell division, when the cell should divide. It coordinates the rate of cell division. What is the rate of cell division? That is also controlled by these factors. Now, not all the cells can have the same cell cycle. All the cells have different cycles, and they are at different stage of the cell cycle. Let us come to frequency of the cell division. So the frequency of the cell division is also maintained by the control of the cell division. So frequency of cell division varies by cell types. Embryo's cell cycle is of 20 minutes. The cell divides at very fast speed. In skin cells, they divide frequently throughout life. 12 to 24 hours is needed for a completion of the cycle. You know, the skin is always exposed to various stress factors and various things. So it is, or wounds or several things. So the skin cells always divide at very faster rate. The damaged cells are removed and they are replaced by the new cells. So that here in the case of skin cells also, the rate of division is 12 to 24 hours cycle. We know that all the night creams are applied during the night. At night, what happens? The cell divide the repair of cell takes place and the division of the cell takes place. Within 24 hours, the cell can divide in the case of skin. Now, in the case of liver cell, they retain ability to divide, but keep it reserved. They divide once every year or two. So they have ability to divide, but the rate of division is very slow and they are at silent state. We will be studying at later on in the later slides. Okay, what happens? Or they are in G0 state, but they can come to division state when it is required. So it is normally every year, once or twice a year. Now, let us come to nerve and muscle cell. Mature nerve cell and muscle cells do not divide at, at all after maturity. Or when once the growth is complete, they do not divide. Nerves and muscle cells, their number is fixed. And so they are supposed always to be in G0 phase. G0 phase is the phase when the cell is in silent state. It is not divided. It doesn't divide. It just grows like that. So this is showing the cell cycle, and see this is the state of division: prophase, metaphase, anaphase, telophase, and then this is cytokinases. Now this whole is part of the cell cycle is known as interphase. We have discussed earlier also. So this is G1 phase, this is S phase, and this is G2 phase. And later on we will describe where are the checkposts for this, what are controlling the division of the cell. Now, so this slide is showing the overview of the cell cycle. There are two irreversible points in cell cycle. One is replication of the genetic material, which takes place in S phase, and secondly, separation of the sister chromatids. If the sister chromatids are separated and they move to two poles, and this stage is also irreversible, and also the replication of genetic material. These two stages are the irreversible points in the cell cycle. So if the replication or if the genetic material, if the check is not proper, and if there is some anomaly in the replication of the genetic material, then genetic aberration takes place. Similarly, if there is when the sister chromatids are not attached to the kinetochore, are not attached or properly to the centromere, then also this problem takes place, or one of the movement of the chromosomes to both the poles becomes unequal, and the daughter cells get unequal number of chromosomes, and that may also lead to either death of the embryo, or it may lead to some genetic abnormality. So there are checkpoints. Process is assessed and possibly halted. So at these checkpoints, because these stages are irreversible, and that might lead to havoc, either the death of the embryo, or there might be some genetic aberration. So as to check this, there are several checkpoints. So process is assessed and possibly halted if there is some problem. We can see this. This is the centromere. These two are single-stranded chromosomes, this and this, and how they are synthesizing their replica and how they separate. Now, checkpoints of the control system. So checkpoint in the control systems are following. What checkpoints do? Cell cycle is controlled by stop and go. Either the cycle will be stopped at that stage only, or it will be continued. Or this is controlled by chemical signals at critical points. Signals indicate if key cellular processes have been completed correctly. So these signals, what do? They check whether the key cellular processes have been completed correctly or not, whether the points, then only they cross the irreversible points. So there are stop and go mechanism at the checkpoints. Now, checkpoints control system. There are three major checkpoints: here, here, and here. So one first point checkpoint is G1S phase, which decides can DNA synthesis begin or not. It is at this stage. So what happens, as I have discussed in the earlier classes, that after the cell division, the daughter cells, they start growing and become metabolically active. Various enzymes and organelles, they are all grown properly, and they are so that they can go for the cell division. So this is the point which is known as G1S checkpoint. Hmm. This is this decides whether it should go to next step or it should be restricted here. Later on I'll go in details what happens at this point. Now G2M phase. G2M phase is this. So what happens at this? A signal from outside the cell comes. Later on we'll be discussing what does that growth signal does, which comes from the outer part, from the outside of the cell. And so this point decides whether whether is this the proper stage that cell should divide, because whether the cytoplasmic material is enough so that it can be divided into two cells. So at this, whether the cell should divide or not, enough metabolic processes have gone, all the enzymes, all the proteins which are required in the cell division are grown in this stage. So if they are not sufficient, they are stopped here, and if they are sufficient, they continue. So at this checkpoint, it is decided whether the cell should go for division or not, or it should enter the S phase or not. So second point is at G2M. Has DNA synthesis been completed correctly, or whether the commitment to mitosis is proper or not. Cell is committed to undergo mitosis or not is decided here. So if G1S checkpoint is crossed, then cell undergoes the synthesis phase or S phase. And what happens here? Here the genetic material or chromosome is replicated. So the DNA synthesizes its own replica. So so that it can divide into two same type of daughter chromosomes, two same type of daughter cells. So once at this checkpoint is crossed, the DNA replication takes place at S phase. Now, so here the DNA replication has taken place. Now, at this checkpoint, which is in the G2 phase, it is decided whether the DNA synthesis has been completed correctly. If not, then it won't proceed further for the mitosis. The DNA is then corrected. It undergoes correction. And there are several things which will be deciding later on. So because if the DNA synthesis has taken place or some mutation has taken place, if it has not replicated correctly, then the cell has to correct its anomaly. So At this point, it is decided whether the cell is committed to mitosis or not, or whether the DNA has replicated correctly or not. If it is incorrect, it is stopped here and cell has to then repair the DNA. Now, if the DNA is repaired, okay, fine. But if it is not repaired, then it will be gone. It will be or it may come to silent stage, or if it will or silent state, which is known as G0 phase. Or if it is required, even cell can undergo apoptosis or die. And that is also genetically programmed. So if this stage is crossed, that is, if the DNA is synthesized completely correctly, then it goes to the mitosis starts. Now there is a third spindle checkpoint. Third checkpoint also, which is known as spindle checkpoint. Are all chromosomes attached to spindle properly or not? Can sister chromatid separate correctly or not? That is very important. This point is also irreversible, because the sister chromatids separate. One cell moves to one side of the pole, another moves to another side of the pole. So that will be proper if it is attached to centromere properly. If the attachment is not proper, then there will be unequal distribution of the chromosomes. One cell can get 2 chromosomes, the another can be deficit of that particular chromosome. So at this checkpoint, if the attachment of the centromere with the kinetochore is not proper, then here also it can stop. The cell division can stop. So this is the third checkpoint. So let us first discuss about the first checkpoint, or G1S phase. So this is the first checkpoint, which is between G1 phase and S phase. Or at this checkpoint, it is decided whether the DNA should synthesize its replica or not. Or this is the first checkpoint for the mitosis, for the cell division. If it is not fit, it will be stopped here only. So G1S checkpoint is most critical, and it is the primary decision point, or it is known as restriction point. If cell receives go signal, it divides. Internal signals are now. At this point there are two types of signals. This decision is taken by two types of signals. One is internal signal, which is actually cell growth, size, its nutrition, because whole cytoplasmic material is to be divided equally into two cells. So there should be proper growth of the cytoplasmic material. So all the enzymes which are involved in the cell division and all the proteins, they should be proper. Their quantity should be whatever should be that which is required for the cell division. That is the cell nutrition. If it is okay, if it's proper, then according to internal signal, the signal is go. Okay. Now external signal is also there, which is basically it comes from outside the cell, from another cell, and it is known as growth factor, which I'll be discussing later on in details. Now, if cell does not receive signal, it exits cycle and switches to G0 phase. So if the cell does not receive the go signal or OK signal from both internal signal system and the external signal system, then the cell switches to G0 phase, and which is G0 phase is the non-dividing or non-dividing working state. Now, let us come to G0 phase. So this is G0 phase. This is non-dividing phase, differential state. Now, most human cells are in G0 phase. Liver cells, no. This is G0 phase or in resting state. This is mitosis, G1, and here the cell has come to, it has not got signal to proceed, so the cell comes to G0 phase. G0 phase, let us discuss the G0 phase. It is non-dividing and differential state. Most human cells are in G0 phase. As for example, liver cell are in G0 but can be called back to cell cycle by external cues. So liver cells are most of the time in G0 phase, but when the cell division is required or the cell gets external signal, they can undergo cell division and or they can become they can proceed and they can come back from the G0 phase to normal phase, normal division phase. Now let us come to nerve and muscle cells. They are highly specialized and arrested in G0 and can never divide. So nerve cell and muscle cells, their number once they come to maturity is fixed. They are highly specialized and they are arrested at the G0 phase and can never divide. See, so G0 phase. Now, activation of the cell division. How do cells know when to divide? Cells communication system is maybe known as the system of the signals, or there are two types of signals which control the cell division. Or cell communication system is managed by the signals which are in the form of proteins. Or these signals are basically chemicals in the living system. In the cells, the signal is in the form of chemicals and they are basically proteins. Actually, proteins are the instruments. They are either activators or inhibitors. So M, once it gets the signal, it comes to the F. Yes, I was talking about the signals. So let us discuss about go-ahead signals. Now, signals are mainly proteins. Protein signals that promote cell growth and division. So there are two types of signals, internal signals and external signals. Internal signals are the promoting factors, internal promoting factor, and which I will be discussing in details later on. And the external signals are growth factors, which are received by the cell from the other cells. Now, primary mechanism of control. So what is the mechanism of control is that all these signals promote basically the phenomena of phosphorylation. And phosphorylation actually is induced by the kinase enzymes. So these kinase enzymes, the phosphorylation of these enzymes, they activate the enzymes, and the kinase enzymes are activated. They are either activated or inactivated. So in go signals, go ahead signals, they are activated. Actually, the enzymes are already present in the cytoplasm, but they come into active form. They are present in inactive form once they are phosphorylated, and this is done by the kinase enzymes. Now, the cell cycle signals, let us talk more about it. So, these signals actually control the cell cycle. Cell cycles controlled are governed by these signals, and they are in the form of cyclins, CDKs, and CDK. These are the steps, CDK cyclin complex. Now, let us discuss about cyclins. They are basically regulatory proteins, and they control the level of the cycle in the cell. CDKs are the cyclin dependent kinases. Now they phosphorylates cellular proteins, and thus it activates or inactivates protein. So if they are phosphorylated with the help of kinases, they become the active form. We get the active form of the enzyme, and which may lead to cell division. So all those enzymes which are required for the DNA replication and further, they are already present but in inactive form. They should be activated with the help of these cyclins and resulting into activation of cyclin dependent kinase, and these kinases, they phosphorylates the protein or enzymes which are involved in the DNA replication and other activities related to cell division. Now CDK cyclin complex. So once the cyclin binds with the kinase, it forms the inactive CDK forms the activated CDK, and they form CDK, or CDK means cyclin dependent kinases. So these kinases are basically enzymes, and which are present in the inactive form. Once they bind with the cyclins, they are converted into the active form, and this is the active form. And this is the once they bind, they form the CDK cyclin complex, which triggers passage through different stages of cell cycle. Now these are the three scientists who involved in these studies, or Leland H. Hartwell. He studied about the checkpoints. Tim Hunt has studied about CDKs, or cyclin dependent kinases, and Sir Paul Nurse has studied about the cyclins. Now let us come to the mechanism. Cyclin and cyclin dependent kinases. Cyclin and cyclin dependent kinases, I am discussing about this, and cyclin drives CDKs and cyclin drive cell from one phase to the next in the cell cycle. So these are CDKs and these are the cyclins. That lead to proper regulation of cell cycle and is so key to life that the genes for these regulatory proteins have been highly conserved through the evolution. So genes which are controlling these two or controlling these points are highly conserved during the evolution, and so these regulatory proteins are highly conserved and they are transmitted from one cell to another to the offspring properly. Now secondly, the genes are basically the same in yeast, insects, plants and animals including humans. So let us discuss about the signals. These are the external signals which we were always discussing about the growth factors, and they are from the cells which are surrounding the cell in question. So they are known as growth factors, and they are about the coordination between the cells. Secondly, protein signals are always released by the body cells that stimulate other cells to divide. So if the cells are very close to each other, then what happens? This G factor is reduced and it is not sufficient to give signal to the cell in question. So there are two types of inhibition. One is the density dependent inhibition, means when the crowded cells are overcrowded, they stop dividing because each cell binds a bit of growth factor, and when they are not sufficient and they don't have enough activator left to trigger division into another cell, then the G factor is not released or the cell in question doesn't divide. So there is anchorage dependence of the cell. To divide cells, they must be attached to a substrate or touch sensor receptors, and that should come from the other cells. This is about the control of cell cycle and uh, control of cell cycle A. And in the next class, in the control of cell cycle B, I will discuss about these factors in details and how do they, in their uncontrolled nature, result into development of the cancer. So I am ending here, and this is about the control of cell cycle, and this is A. I will be discussing in two classes. This is control of cell cycle A, and in the next class I will again go in details, and that is control of cell cycle B.